So good day everyone, I'm Alisa M. Agustin and I will be reporting the second subtopic of Virology, which is the virus-host interaction or the virus infection. So before we move on to our discussion, let's have our mini game. So this mini game is entitled Name the Virus. You will guess or name the virus that I will be showing you in a form of pictures. So we have here picture number 1, picture number 2, and picture number 3. So if your answer on picture number 1 is rubella virus, you were correct. And if your answer on the second picture is human immunodeficiency virus, you were also correct. And if your answer on the third picture is rabies virus, you were also correct. So good job! So yeah, over in there. So we are done on our mini game, so thank you for participating. So here is our topic outline. I know it's a lot, but bear with me. I will do my best to discuss this very well and in a way that you will understand it. So let's move on. So first is the virus infection. Virus infection occurs when a non-living virus enters the body, attaches to the host cell, and hijacks its machinery to make more virus copies, which then spread and infect other cells. So basically, virus infection is when a virus gets to enter in our body, it infects the host cell, and then it's spread out. When it is spread out, it infects other cells, and when it done that, it can spread throughout our body that can cause virus infection. So we are done in identifying what is virus infection. Let's move on to the next slide. So next is the viral infection expressed in host cell. When virus infect host cell, they can cause different effects, from no visible damage to complete cell destruction. The type of cell depends on the virus and the host response. For example, a good example of this is varicella zoster virus that can cause chickenpox. If the person have low immune system, the symptoms of chickenpox is a lot worse than a typical, and it can also lead to having a pneumonia. But if the person have strong immune system, if the person have strong immune system, the symptoms of the chickenpox can be milder than the usual. So that is how the effect depends on the virus and the host response. So let's move on to the next slide. So these are the types of viral effects. Number 1, we have the cytocidal or the cytolysis. It causes cell death. Again, a given example we have this is the polio virus. So polio virus can cause what we call polio, and this polio virus attacks the nervous system and it causes paralysis. That is why in cases we saw on people having polio, they have paralyzed legs. It can be left or right, or in worst case scenarios, it can be both legs. So number 2, we have cell proliferation. Leads to abnormal cell growth. So again, a given example we have here is molluscum contagiosum. So this is a type of virus that infects the skin. So it forms bumps, warts that is reddish. So the common name of this molluscum contagiosum is water warts. So it is abnormal cell growth. So it forms little warts in our skin. So number 3, we have malignant transformation. So this converts normal cell into cancerous cell. Given example we have this is oncogenic viruses. So oncogenic viruses can cause or can transform normal cell into cancerous cell. So a good example of this is human papillomavirus that can cause cervical cancer. Another example is T-cell leukemia type 1 that can cause T-cell leukemia or cancer in the blood, specifically in white blood cell. And another good example is hepatitis B virus and hepatitis C virus that can cause liver cancer. So let's move on to the next slides for us to know what are the other types of viral effects. So here are the latent effects. Virus remains dormant and may reactive Given example, we have herpes simplex virus. So this kind of virus are hidden, they hide in the cell. They remain dormant, meaning when they are dormant, they are not multiplying. But once they are reactive, or once they begin to be active by some stimuli, they can be active and can cause their typical symptoms. For example, in herpes simplex virus, that can cause genital herpes or common colds, something like that. So number 5, we have steady state infection. So virus persist without causing injury to the cell. So number 6, we have morphological changes. Infected cells form inclusion bodies. A given example we have here is the rabies virus. So morphological changes. So it shows visible changes on the cell. That is what it means. So in a form of inclusion bodies, and later on we will talk more about inclusion bodies. So let's move on to the next slide. So we are done on our second topic outline. So now we are ready to know the mechanisms of viral-induced cellular injury. Viruses cause cell damage through different mechanisms depending on the type of the virus. So meaning, if we have this virus and this virus, we have virus 1 and virus B, they have different mechanisms to infect the host cell. So we are here on the main mechanisms of how the virus causes injury to the host cell. So the first is the inhibition of the host synthesis. Viral proteins stuff DNA and protein production of the host cell. So first, ang ginagawa ng virus ay i-stop niya yung main production ng DNA and protein ng host cell. Second, we have viral accumulation. Build-up of viral materials distorts cell structure and becomes toxic. So, the first one is i-stop niya muna yung DNA and protein synthesis ng host cell. And then, after that, magte-takes over na siya. Magbi-build up na siya ng sarili niyang material. Meaning, magsisynthesize na siya ng viral DNA niya, ng viral protein niya, na yun na yung mismong magiging materials ng cell. And it can cause destruction and pwedeng maging toxic yung loob ng host cell natin. Number 3, we have membrane permeability changes. It alters the cell membranes, causing enzyme release and the cell destruction. So once na mag-accumulate na yung viral or mag-spread na yung viral material sa loob ng host cell, magkakaroon na ng changes that alters the cell membrane. It can cause enzyme release. And once the enzyme release, it can cause cell destruction or autolysis. Meaning, masisira na yung mismong cell dahil sa enzyme release ng mismong cell na yun. Dahil na-alter na nga ang cell membrane. Next, we have the number 4. Cytoplasmic membrane alteration leads to cell fission or syncytium formation or new antigen expressed on the cell surface. So, once na mag-accumulate na yung viral infection, nag-spread na siya sa loob ng cell, it can lead to new antigen expression on the cell surface. This can lead to syncytium formation. For example, we have here infected cell, and we have here uninfected cell, infected cell, infected cell, and infected cell. Once they are close to each other, they can fuse, and this can cause syncytium, meaning magiging isang malaking cell na lang sila na madaming pinagsama-samang cell. So that is how one of the mechanism of how this viral can cause cellular injury. Number 5, we have chromosomal damage. Certain virus, for example, mumps, measles, and adenovirus causes genetic injury to host chromosome. So one of the important genetic information is on the chromosome. So these viruses attacks these chromosomes and make chromosomal damage. So number 6, we have the inclusion body formation. Abnormal structures appears inside the infected cell, indicating viral infection. One of the indicator that there is a viral infection in the host cell is the formation of inclusion bodies. As I said earlier, these inclusion bodies are visible changes, or in the types of effects this can cause morphological changes in our host cell. So I will be discussing it mamaya-maya lang. Malalaman natin yan. So let's move on. So we are done on the mechanism of viral induced cellular injury. So we already know how viruses processes on how they infect or they damage a single cell or the infected cell. So next is the inclusion bodies. So I have said earlier, we will dig in deeper on what is inclusion bodies. So these are globular clump or masses that forms inside the infected cell when viruses multiply. So aside from that, inclusion bodies are very useful in identifying what kind of virus infected the cell, because each type has specific location and has specific appearance. Mamaya malalaman natin yan. So we have here our 3 types of inclusion bodies. So first we have cytoplasmic inclusion bodies. So basically these are found in the cytoplasm of the cell and usually made by RNA viruses. Give an example, we have Negri bodies in rabies. So Negri bodies is basically the inclusion bodies called that the rabies virus formed in the host cell. So cytoplasmic inclusion ay nasa cytoplasm lang siya. So next we have intranuclear inclusions. So these are found inside the nucleus and commonly formed by DNA viruses. Give an example, we have Cowdry type A in herpes virus. So this intranuclear inclusion can be found inside the nucleus or sa nucleus lang siya. And number 3, we have both cytoplasmic and nuclear inclusion. So pag sinabing ganito, this inclusion can be both found in the cytoplasm and inside the nucleus. Give an example, we have measles virus. They can form both types of this at the same time. So I will be showing you pictures for you to be able to illustrate it in your mind. So we have here on our first picture, we have the cytoplasmic inclusion. So we have here the cell, and as you know, and our inclusion bodies are the pink side, or our virus are the pink one. So the inclusion bodies are only on the cytoplasm, and on the other side we have the intranuclear inclusions. So makikita nyo naman yung pinagkaiba, yung intranuclear inclusion bodies natin. The inclusion bodies are inside the nucleus, and there are no inclusion bodies on the cytoplasm. But in the middle picture, we can see this in both, which is the cytoplasmic and intranuclear body inclusion. So we can see this, that the inclusion bodies are both on the cytoplasm and inside the nucleus. So that are the types of inclusion bodies that can be found inside the cell once they were infected. So inclusion bodies can be seen under a microscope using special stains. So sabi nga natin kanina, inclusion bodies are very important in identifying viruses. Malalaman natin if what kind of virus is that, because of their inclusion bodies, it has a specific location and it has its own appearance. So acidophilic inclusions, so there are kinds of inclusion bodies that it can be acidophilic and basophilic. Acidophilic inclusion stains with Giemsa, stain pink with Giemsa and eosin, and basophilic inclusion stain blue with hematoxylin. So acidophilic inclusion bodies stain pink, and this acidophilic meaning they can bind on a stain that is acidic, and in basophilic inclusion they stain blue because they can attach to basic solution stain, something like that. one is acidic, and one is basic. So we have here inclusion bodies in red blood cell, and these are both cytoplasmic inclusion, because we know in matured red blood cell it has no nucleus. So these are both cytoplasmic inclusion, but we have here different stain. One is a basophilic inclusion and one is acidophilic inclusion. So let's move on to the next slide, because you already know what is inclusion bodies, the types of inclusion bodies, and how we can stain them. So we are done in our inclusion bodies. So let's move on to the next slide. So now is the route by which viruses enters the host. So these are the routes, or ito na yung mga ruta ng viruses kung paano sila pumapasok sa katawan natin. And dito, this is one of the most important, because this is how the virus starts the infection. Dito na kasi sila papasok. So let's talk about this. So let's move on to the next slide. So first we have the mucous membrane. So we have here the conjunctival sac. The entry point is through our eyes. Next we have our nose and throat, which is the respiratory tract. Enters by inhalation. And next is the gastrointestinal tract. Enters through ingestion. Number 4, we have genital tract. Entry during sexual contact. So we are done on the mucous membrane. So let's move on to the next slide. Next is the skin. Through abrasion or cuts on our skin, papillomavirus can enter. Through insect bites, arbovirus can enter. Through animal bite, virus or rabies virus can enter. Through injection or needles, hepatitis B virus and HIV virus can enter our body. So next we have the subcutaneous and muscular route. Viruses enters deeply through insect bites, animal bites, and injections. A given example, we have rabies virus and arbovirus. So subcutaneous route, parang katuluyan lang to ng route sa skin, because once na mag-enter sa skin natin yung virus, it can dig deeply to our subcutaneous or the muscles under our skin. So we are already done on the mucous membrane, we are done on the skin, we are done on the subcutaneous and muscular. Now we are dealing with placental route or the vertical transmission. Viruses crosses the placenta from the mother to fetus. Given example, we have the rubella virus. So it is sad because normally the placenta acts as a strong barrier that protects the baby from the infection. However, some viruses are strong enough that it crosses it and it can cause infection to the baby that can cause complications, and it is sad. This torch infection are the viruses and bacteria, parasites, that are the exemption on how the placenta protects the baby from the outside world. So we have here the T, meaning the Toxoplasma gondii, the other agent, which is the syphilis, parvovirus B19, the varicella zoster virus, the listeria, R we have rubella, C we have cytomegalovirus, and H we have herpes simplex virus 2 or the HSV2. So these torch infections are vertically transmitted via placenta or via blood, body fluid, or breast milk. Let's move on to the next slide. So we are done on the routes by which viruses enters the host. So we have routes. So we have different routes including membrane mucous, skin, the subcutaneous or muscular route, and the placental route or the vertical transmission. So let's move on to the next slide. So spread of virus inside the host cell. After entering the body, viruses may cause local infection and general infection. So local infection stays on one area, like cold on our nose. So in general, infection, or also called as systemic infection, is spread through our blood, lymph, or nerve that affect other organs like measles and chickenpox. So localized infection on one area, and in general infection, these viruses on local areas spread out through our body. So let's move on to the next slide. So we have here spread of viruses inside the host cell. So first we have direct cell-to-cell spread. The virus moves from one cell to nearby cells through small bridges between them, seen in warts, common colds, and eye infection. So next we have through macrophages. Some viruses survive inside macrophages. So basically these macrophages are immune cell that supposed to be eating or cleaning up these viruses in our body. But some of the viruses are clever, kaya nakaka-survive sila sa loob ng macrophages, and instead na mamatay sila sa loob ng macrophages, ginagamit pa ngayon nila yung macrophages na yun para mag-spread throughout the body. So it is sad. So number 3, we have through lymphatic system. After entering the skin or mucous membrane, the virus goes to lymph nodes. If not stopped there, it spread through the lymph and enters the blood, reaching the organs. And next, once they are spread on the lymphatic system, they are now in the blood. So through the blood cell, through blood and vessels, called the viremia. So virus first appears in blood as primary viremia, then multiplies in liver or spleen, causing secondary viremia. So it can travel inside blood cells or freely on the plasma. So next we have through the nerve fibers. Some viruses move along nerves to reach other part of the body or the targeted part of the body. Give an example here is we have herpes virus, Rivas virus, and the arbovirus caused by insect. So through nerve fibers is they use these nerve fibers for them or for this virus to reach the targeted organ. So for you to visualize this spread of viruses inside the body, which is the cell-to-cell, in macrophages, in lymphatic system, in blood and vessels, which can cause viremia, and in nerve cell, I will be showing you pictures of this for you to better illustrate it in your mind. So we have here the direct cell-to-cell spread. So we have here the infected cell, and this infected cell can cause destruction to neighboring healthy cell. So this infected cell can cause or can spread the virus through the neighboring cell, causing it to spread the virus. Next, we have through macrophages. So we have here in our illustration the large macrophages or our white blood cell. These macrophages should be eating or phagocyting these viruses, but some viruses are strong enough to survive inside the macrophages. So while they're inside the macrophages, these viruses use these macrophages to travel along the blood. So once they're on the blood, it can enter the lymphatic system. So in mucus or in skin, when the virus enters the skin or through mucous membrane, it can reach the lymph node. And if the viruses are not stopped on the lymph node, it can travel to lymphatic vessel. And once they're in lymphatic vessel, their next route is on the bloodstream. Once they're on the bloodstream, they can spread through blood and vessel, causing primary viremia. And once this viremia reaches the liver or spleen, they multiply inside these organs. And once they multiply inside this organ, they will they will be released in a larger portion, causing secondary viremia that can reach the targeted organ. So this through nerve fibers. So we have here an illustration. We have here our skin and mucous membrane. We have cuts here on the skin, and we have virus enters that. It uses nerve fiber. It moves through this nerve fiber. Virus move along nerve fiber until it gets to its targeted organ. We are done on the spread of virus inside the host cell. We identify how viruses spread inside our body via cell to cell, by macrophages, via lymph node, via blood and vessels, and via nerve cells. Now we are ready for viral tropism. So viral tropism means the specific attraction or preference of a virus for a certain organ or tissue in the body. So each virus affects only a particular parts depending on where it can attach, enter, and multiply. So may pagka-choose yung mga virus natin sa viral tropism. Sa viral tropism, meaning yung virus may favorite place sila na pupuntahan, or yun lang yung kaya nilang i-infect. And later on, we will know and understand what viral tropism is. So, this we have common target organs include the skin and central nervous system. But viruses can also affect the liver, lungs, muscles, mucous membranes, and our heart. So, based on the organ they attack, viruses are grouped as dermotropic, or yung favorite place nila. Sabihin natin favorite place nila, yung gusto nilang i-infect, is the skin. The neurotropic virus, yung favorite place nila is nervous system. Pneumotropic, yung favorite place nila is of course the lungs. Enterotropic, favorite place nilang infect is our intestine or yung gut natin. So, heterotropic, our last one, based on the group, ang favorite place nila is the liver. So, on this picture, we can visualize what is viral tropism. For example, in cellular tropism, meaning the virus choose a specific cell to infect. HIV normally infect macrophages but not neurons. So that is what we call yun nga yung mga favorite nila. O yung specific, yung prefer nilang i-infect. So HIV infect macrophages but not neurons or brain cells. So in tissue tropism, influenza virus normally infect lung tissue but not brain tissue. Again, influenza virus, tissue tropism, they infect lung tissue, not brain tissue. Kaya, ang effect ng influenza ay dito banda sa ating lungs. In host tropism, mycosomovirus, what do you think it infects? The rabbit or the human? Okay? Mycosomovirus normally infect rabbits, but not humans. So, this virus cannot infect human, meaning, if this virus enters the body, it cannot put any harm on that person because host tropism, it only prefers rabbits. So that is viral tropism. So next, we are done on the viral tropism. We have incubation period in viral infection. The incubation period is the time between the entry of the virus into the body and the first appearance of the symptoms. So during these times, the virus already multiply. It already multiply and spread in the targeted organ before causing the illness. So imagine that, bago pa tayo makaramdam ng symptoms ng virus na yon, it already multiply and spread out through our body before having those symptoms. So is that clear? Clear! So we are done on the incubation period of viral infection. So let's move on to the next slide, the release of virus from the host cell. So the release of virus is the final stage of the process of infection. So the viruses are usually released in the few days of the incubation period, during the acute stage of the illness, and only for a short time after recovery. So we are done on the release of virus from the host cell. Now we are ready to the next topic outline, which is the host response to viral infection. So in this slide, we have the host response to viral infection. An immune response is the body's first line of defense against harmful invaders like bacteria, viruses, and toxins. So, hindi lang nagre-respond yung immune system natin sa viruses, but they can also respond to bacteria, parasites, and so on and so forth. So, our immune system is our natural defense against these bacteria, viruses, and toxins. There are two types of immune response, the specific immune response and the non-specific immune response. So the non-specific immune response is the body's first line defense. It attacks any invaders in a general way. So this non-specific immune response, meaning they don't have a specific target or they don't have specific targeted bacteria or viruses. Basta nag-act lang yung immune response natin na to pag merong foreign bodies na nag-enter sa body natin. Next is the specific immune response. This specific immune response is specifically called specific immune response because they were specific. We have here specific and non-specific. So let's move on to the next slide. So we have here our specific immune response, and one of this is called antibody-mediated immunity or the humoral immunity. One of the example in the bullets we have immunoglobulin G and immunoglobulin M. Protects blood and tissue from the viruses. Meaning yung immunoglobulin G and immunoglobulin M can be seen on our body and our tissue, and they fights against viruses that resides on that blood and on that tissue. So next, on the next bullet we have immunoglobulin A, found in mucous, respiratory, intestinal, and genital tract. Blocks virus entry and at local site. So this immunoglobulin A stop the virus in the entry points. Ibig sabihin, hindi pa sila totally nakakapasok or naka-infect sa body natin. Ini-stop na sila ng what we call, or na-identify na sila ng what we call the IG or immunoglobulin A. Next, we have another specific immune response is the cell-mediated immunity or the CMI. The T lymphocyte or cytotoxic T cell kills virus-infected cell. Next, we have NK or natural killer cell. And killer cells destroys infected cells directly. So like the T lymphocytes, NK and killer cell also destroy the infected cell. Macrophages help limit virus to spread. Interferons from T cells stop virus multiplication. So later on, we'll talk more about interferons. So next is the non-specific response. So ito na nga yung sinasabi natin kanina na they don't have specific targeted viruses or bacteria. Instead, they fight any other foreign bodies that enters the body. First is the phagocytosis via macrophages. Macrophages clear viruses from the blood. So aside from being specific immune response, macrophages is also part pala ng non-specific immune response. Next is the body temperature. High temperature above 90, Body temperature, high temperature above 39 degrees Celsius shows down virus growth. So, a good example of this is fever. Pag nilalagnat tayo, we often experience body high temperature. And that is a good example of our body's immune response. Meaning, pinapainit ng katawan natin yung mismong katawan natin para i-fight yung certain viruses and bacteria na yun. And we all know that some viruses and bacteria, in their morphology, they are very sensitive. Some of them are very sensitive in high heat, higher than 39 degrees Celsius because their optimal temperature is what? 37 degrees Celsius. So next, our non-specific immune response is what we have also, the interferons or IFN. Natural antiviral proteins. So these are type of proteins, which is antivirals. Antiviral, or they can help by stop the spread of the virus. So we will talk more about interferons later on. So we are done on the host response to viral infections. Now we are ready for the interferons. So as we all know, the interferons are a non-specific response and also a specific response of our immune response. So there are different kinds of interferons, and again, interferons are antiviral protein. They are protein. We have alpha interferon, beta interferon, and gamma interferon. So interferons doesn't directly kill the virus. It is released by the infected cell and acts on the nearby healthy cell. It binds to receptor on those cells and activate protective enzyme. Again, for us to better illustrate this in our mind, I have this picture. So as it have said on the slide earlier, the interferons are released by the infected cell. These proteins are released in the infected cell, then travel to the neighboring healthy protein. And these interferons enter the healthy proteins via its receptor on the plasma membrane, of course. And once they enter the cell, it signals the cell that activates specific enzyme. As a result, neighboring cell becomes resistant to infection and the spread of the virus is slowed or definitely stopped. So that is how the interferon helps neighboring cell or healthy cell via the signal of the infected cell. So ang galing, ano? So we have here the agent that induces interferon production. We have weak inducers, which is the cytocidal and virulent viruses. The strong inducer, we have virulent viruses. RNA virus induces more interferon than DNA viruses. Double-stranded RNA, can be natural or synthetic, is very effective inducer of interferon also. So next, we have other inducers. Animal, plant viruses, intracellular organisms such as bacteria, rickettsia, and protozoa. And in bacterial products such as lipopolysaccharides and polysaccharides. So we all know that interferons are non-specific immune response and a type of specific immune response. And immune response can also respond to bacteria and protozoa. And these interferons can also respond to bacterial products such as what we have here, the lipopolysaccharides and polysaccharides. So that are the inducers of interferons. Now we are ready to talk about the biological effects and uses of interferons. So, biological effects of interferons includes antiviral activity. It stops virus replication. Antimicrobial activity acts against intracellular microbes like Toxoplasma and Chlamydia. Immunoregulatory effects activates macrophages. So this interferons pala can activate macrophages that can undergo phagocytosis or engulf these viruses or bacteria that enters the body. So NK cells and T cells. NK cells stands for natural killer cells. And T cells regulates antibody production. So interferons is also an anti-tumor activity. It slows or stops tumor growth. And we have here the last bullet, which is the effects of interferons includes cellular effects, inhibits cell growth, DNA protein synthesis, and increase MHC antigen expression. So now we are done on knowing the biological effects of interferons. Now we are ready to know the uses of interferons. So interferons are used in the treatment of viral infection and benign tumor and in malignant tumor. So we are done, and now we are on the immunoprophylaxis of viral diseases. So because only few antiviral drugs exist, prevention through vaccine is the best protection. So there are only a few drugs that can treat viral infection, so it's hard to cure viruses once you get them. Vaccine teach the body to recognize and fight viruses before you get sick, making them the best way to prevent viral diseases. So sabi nga nila, prevention is always better than cure, diba? So we have here our vaccine or the immunoprophylaxis. Types of vaccine includes live vaccines. We have here types of vaccine. First is the live vaccine. Contains weakened, attenuated viruses that causes mild infection but gives strong, long-lasting immunity. Number 2, we have killed or inactive vaccines. Contains dead viruses, safer but provide weak and shorter protection. Live vaccine contains live virus, and the killed vaccine contains dead virus. The live vaccine have a long-lasting the live vaccine can provide long-lasting and strong immunity, but the killed vaccine can provide weaker and shorter protection. Let's move on to the next slide. Next, we have subunit vaccine. It contains specific viral antigens that triggers immunity without using the whole virus. And we have number 4, recombinant vaccine, made using DNA technology to produce viral antigen in lab organisms like E. coli. And 3, we have subunit vaccine. It uses parts of that virus that can trigger the immunity. or that promotes immunity. And we have this recombinant vaccine that is lab-made for us to have our immunity. Next, we have passive immunization. So this gives temporary protection using ready-made antibodies from human serum or gamma globulin. So passive immunization provides immediate but temporary protection by giving a person a ready-made antibodies from other person or animal. So a good example of this is when we got bitten by a dog. Ina-advise agad nila na pumunta tayo sa pinakamalapit na center para makapag-take ng rabies vaccine. So that rabies vaccine is a passive immunization, meaning it has rabies antibodies para i-fight nung antibodies na yon yung rabies virus na pwedeng mag-spread out through our body to its targeted organ, which is the brain. Kaya it is very advisable once you got bitten by a dog, cat, or something animal, you are advised to take that rabies shot or rabies vaccine, because in that way you can get temporary protection using that ready-made antibodies by those passive immunization. We are ready to move on to our last topic outline, which is how viral infection are stopped via the drug treatments or the treatments. Here are our first two drugs that inhibits the attachments and penetration, which is the first step of the replication process. We have here amantadine that blocks influenza virus from attaching and entering the cells. We have ramantadine, similar to amantadine, but with fewer side effects. So these two drugs, amantadine and ramantadine, are specifically antiviral drugs that inhibits the attachment and penetration of that virus on the host cell. So we have inhibitor of viral nucleic acid synthesis, meaning these antiviral drugs are responsible on inhibiting the steps of viral nucleic acid synthesis. We have here the acyclovir, ganciclovir, we have vidarovine, we have here the trifluorothymidine, we have here the foscarnet, 7 we have acetathymidine, 8 we have dideoxyinosine, 9 we have dideoxycytidine, and then we have this ribavudine. So these antiviral drugs works by blocking viruses from making DNA and RNA, which is said here the inhibitor of viral nucleic acid synthesis, which is the diribonucleic acid and deoxyribonucleic acid, DNA and RNA, stopping them from multiplying. Some targeted herpes virus, while others targeted HIV or respiratory hepatitis virus. A few drugs like foscarnet are used for viruses that are resistant to other treatments. So, very fascinating ang mga antiviral drugs na to, na very useful in blocking or inhibiting the viral nucleic acid synthesis of that virus. So next, we have here, I think, our last slide, which is the inhibitors or the antiviral drugs that inhibits the viral protein synthesis. Again, we have antiviral drugs for attachment and penetration. We have antiviral drugs for nucleic acid synthesis. And we have viral drugs for protein synthesis of that specific virus. And one, we have interferons. Interferons are not only proteins, but they're also in a form of drugs. So this interferon blocks translation of viral mRNA used for chronic hepatitis B and C. We have here our second antiviral drugs that inhibits the viral protein synthesis, which is the methaisazone. Stops late viral protein synthesis used for pox virus infections. So those are the different antiviral drugs that can stop specific steps in viral multiplication cycle. So it can stop, so some drugs can stop the virus from penetrating or attachment. Some viral drugs can stop the DNA or the RNA synthesis, and some drugs can prevent or inhibit the protein synthesis of that specific virus. And overall, that is the end of my topic. And thank you for watching. I hope you've learned and you understand what I've been saying right here, because I hope we get better every day. And thank you. Goodbye.